and welcome to the NRPSDesigner. This software was developed by the IGEM team of Heidelberg in 2013. Our project focused on making non-ribosomal peptide synthesis accessible to the synthetic biology community. For this, we actually submitted an RFC 100, which describes the whole pipeline of working with such synthetases. You can read more about it on our wiki. Still, one very important part of this whole framework is the in silico design of appropriate DNA sequences. This, as you can see in the video, is done using the NRPSDesigner. Thus, here I'd like to explain to you how you can use our software in order to create your own NRP constructs. In the main page of the NRPSDesigner, you can either get over this drop-down and choose the NRPSDesigner functionality, or you can get there by pressing the Start Designing button. Once you're here, you need to add your own entry. We'll just name it Test. We'll click on it, and then we get to the core page of the NRPSDesigner. Here, you basically choose the monomers out of which your final peptide should consist. For example, we might want to choose a non-proteinogenic amino acid, such as ornithine, and even in its D configuration. And then we can choose another one. For example, just choose glutamine. While you're doing your selection of monomers, the peptide you're about to create a synthetase for is always shown in this panel on the right. I'd also like you to note this button here, Tag NRP with indigoidine. If you've chosen it, then your final construct will also contain the indigoidine synthetase, which means that your final peptide will be blue and easily detectable. This Use Curated Domains Only button will be explained later. Once you've done your selection, you can press on Design Construct. Then, a request is submitted to our server and an optimal sequence of domains is calculated. This sequence of NRPS domains is of course capable of producing the peptide. Here you can see these domains. The adenylation domains are the ones which confer the specificity to the amino acid of choice. Finally, those three domains are the ones of the indigoidine synthetase, necessary for the tag. But then we also move one layer of biological organization below, to the actual DNA sequences. Here you can see the fragments that have to be assembled together in order to get the functional synthetase. For example, the condensation domain here could be amplified by PCR from the genome of Brevibacillus parabrevis. In order to assemble all of those genes, in our experiments we mainly used Gibson assembly. For this reason, we integrated Gibson into our software. Gibson was developed by the Cambridge IGEM teams of 2010 and 2011 in order to facilitate primer design for this cloning method. What you might have noticed here is that you get the final functional synthetase, but in your final plasmid you probably won't have your own backbone and promoter. This can also be easily done. You just switch app and go to the Manage Gibson fragments. Here you can add a new fragment. Again, this from Gibson, so you can learn more about it on their site, but we can easily add parts from the registry by using the API. For example, if we want to be very standard, we might want to import the PSB1C3 backbone. As you can see, it has been added to your library, and you can quickly see its sequence. Then, if we go back to the designer, your peptide has been saved, so you can click again. Its monomer sequence is still saved, as well as its design, so it's still the same domains. But now you basically can add your backbone to the final construct. Note that you can't put it at any position because our program disallows breaking the structure of the synthetase. Once you have your final construct, you can actually go on with a primer design. Here, the calculation takes some time because also M-fold is used in order to issue warnings depending on different thermodynamical considerations. Here you can see the final output. For example, for the condensation domain that we saw earlier, you would need to use the following forward primer. It basically consists of a 3' sticky end from which the polymerase amplifies, as well as the 5' Gibson overhang. Now, if we move back to our initial peptide choice, you can basically see that we chose D-ornithine and L-glutamine. But if we want to create combinatorial libraries, we might want to be interested to be able to shuffle or to exchange one of those monomers, for example L-glutamine, by other ones, starting with a construct we already created. This feature is also integrated into the designer. Just go back to the main page, and then instead of clicking on test, you need to choose this create new library option. Here you can see again your peptide, but you have the ability of actually adding new ones. For example, we can add L-asparagine or even another one, say serine. Actually, if you press on this one, all monomers in the database will be chosen for this position. Once you've made your choices for a combinatorial library, you can click again on the Design Library button. A request is sent to the server again, and for each of the new monomer combinations, an optimal domain sequence is calculated. This takes some time because our algorithm used the Dijkstra algorithm in order to calculate an optimal sequence based on evolutionary considerations. For example, if two domains are next to each other in their native organism, then you'd probably want to take them together in your final construct. This would for one mean one less PCR amplification, but since you're also not disturbing the native structure, the chances of the final construct working are higher. You get this library table which contains one row for each variant you created, here for asparagine and serine. If you select your variants, you can then also download a detailed description of each one of them. In particular, the description is provided in GenBack and SBOL format, and a PDF of the cloning procedure is also provided. Beyond these, the zip file also contains a detailed file of the primers that have to be ordered in order to get all your constructs. So as can be seen here, this is the main construct, the original one, and then there's the variants you create by modifying the one position. All you have to do is to order the primers and then amplify the genes and clone them using Gibson assembly. So this wraps up one of the important parts of the designer, which is the creation of sequences which code for synthetases, which again can produce your peptide of choice, or your library of peptides. But another very important consideration is that you are currently limited by what is available in our database. Thus, we've also made it very easy for the community to extend our database. For this, just go to the EPS dropdown and choose the Add NRPS into Library function. Here you can enter your DNA sequence. This can be a raw string or a FASTA formatted sequence. I've copied here the sequence of the indigoidine synthetase of Photorhabdus luminescens. Here you would basically choose the organism and also the product. Of course, you can add new organisms or new products. You also need to add the gene name and a short description. Once you're done, you just press the Generate Domain buttons, and then based on a hidden Markov model pipeline we created, based on anti-smesh, actually returns the domain predictions. For example, the first one has been predicted to be an AOXA domain. This is novel to our tool, as other NRPS domain prediction tools can't detect it. Here you can see the domain boundaries automatically predicted. Of course, the indigoidine synthetase also has a thiolation domain, also predicted, and a thioesterase domain. In order to be easily able to compare the domain predictions to other domains in the database, you can get this multiple sequence alignment view, where it is compared against everything in the database. This could actually allow you to choose the boundaries depending on the alignment rather than the hidden Markov model prediction. In the case of adenylation domains, not shown here, you can also choose the substrate specificity. This is also automatically predicted. Once you're done, you can submit it to the server by pressing this button. As you can see, now a sequence was successfully added to the database. Now you might also wonder what happens if any user can just add his own sequences. This is what the curate button you saw in the beginning is all about. This allows one to filter the available domains to the user depending on who actually did the curation for a particular domain. Curated ones are the ones which were entered by us or other NRPS experts. If you also want your domain entries to be curated, you need to request appropriate privileges. You can just go on your profile and click on this button. Once you submit your request, we will try to answer to your email as quickly as possible and provide you with the appropriate rights. This basically wraps up the functionalities of the NRPS designers. So now it is up to you to create your novel non-ribosomal peptides, which could be antibiotics or useful for recycling. We hope that you will find our software useful, and we encourage you to read more about the algorithms and the database on our wiki. Also, we would be very happy if you forked our project or sent us a pull request.